Good morning, everyone. Welcome back to Bioinformatics for Infectious Diseases. We're going to start here shortly. And before we start, I just wanted to remind everyone that the sessions are recorded and we're going to be posting the sessions on the YouTube channel so that you can participate by watching the videos asynchronously. So let's start by talking about where we are as a group. We have a lot to cover today, and so I'm going to try and move quickly through the review. Hopefully, most of you are now getting hands-on practice with sequence alignment and mapping out specific variants, but I'm also hoping that you're starting to understand conceptually how the nucleotide variation, the different types of pathogens, and the process of disease progression inside a single host, as well as separate hosts and the transmission of disease, all play a role in our ability to study the genomic component of infectious diseases and understand how the disease is manifested in different types of diseases. So to understand where we are as a group, we can go to the main page of the Bioinformatics for Infectious Disease organization, or the main page. And what I see is a clear indication that most of you are making a lot of good progress. Remember that we have three components. We have the educational theoretical material, which is some of the materials that we've discussed, like Introduction to Genomics, Genomics 1. We talked about some projects like Spotaptera frugiperta, or the SF9 detection of viral sequences by mapping on a reference database. We also talked a lot about the Ebola virus, and we have a project on the Ebola viruses already uploaded. And then we also talked about some aspects of specific types of diseases, COVID, malaria, etc. So you should have been able to play around with some of those data sets. Another component, so the orange here is the completion of theoretical material. The gray is running those practical pipelines. And then the red is actually the coding practices that we've done. Now, we haven't done too many, but I'm hoping that most of you are getting a good sense of what sequence alignment looks like from a coding perspective. So in R, we had some multiple sequence alignment and pairwise sequence alignment that were important to understand just from a coding standpoint. I did want to mention that all of this information is also stored on your individual profile. So thank you, Mona and Dr. Manav Jain, who have completed, updated their profiles, and we can clearly see their progress. So they're starting to do a lot of the practical assignments, and I'm hoping that this is helpful to you and makes you understand the material better. I know that over the weekends is a good time to catch up for those of you that are still going through the first few sessions, trying to understand how we got to where we are today. And to make sure that we address all of the technical questions, I wanted to highlight some of those things that were posted in the assignment forum. And also, we'll talk a little bit about some of the challenges that you've encountered running those pipelines. So first of all, we had this practical assignment, which Beeps posted on the forum. And by the way, before I get into this, is there anyone here that was not able to understand, what exactly to do, or where to find the data, or where to find the article that we discussed last time? Was there anyone that had trouble finding any of the resources? Again, you're welcome to post your comments and questions in the chat, and that way I will know. If you did not have a problem finding it, then you remember that, okay, so, okay, great. So I think the video recording is also posted on the forum, so you can check that out. And what we did last time was we talked about this publication called Ebola virus glycoprotein with increased infectivity dominated the 2013-2016 epidemic. And we talked about it in the context of mutations that could characterize different strains of viral infection. So we can look at rates of transmission, rates of mortality. We can look at how the virus potentially comes from different animal hosts. So we talked about zoonotic transmission last time. But one of the things that we can also look are the genomic sequences and how we can understand the different lineages or established mutations in the course of an outbreak. And that outbreak could be epidemic, which is localized, or pandemic, as we see now with the coronavirus. So in all of these cases, there are going to be specific strains and lineages of those strains that can help us categorize the different types of sequences that were found. So what they discussed in this article were some analyses that were conducted over the course of this epidemic, and different types of mutations were identified. And so we talked briefly about those mutations. As you remember, mutations could be categorized into different types, and those types include mutations that cause a change in an amino acid. And so today we'll talk a lot more about those mutations, but there are also mutations that do not result in a change in that amino acid, but also there are mutations that cause a stop codon, which typically makes the protein shorter or significantly different from how it is intended to function. So typically, those kinds of stop codon mutations result in a non-functional protein. So you can kind of imagine that those probably would not be beneficial to a virus. And then the ones that change the amino acid to some other amino acid can change different types of properties. And so we'll talk about that a lot. And so Mona, for example, answered here that you can see the impact of how much data is available. For example, we saw last time after we were almost over with the session that there are over 3,000 nucleotide sequences for Ebola overall. But at that time, they only had about 1,500 sequences. And you can see that they were taken and analyzed just the same way that we are analyzing the data to extract mutations that were very significant. So the specific mutation correctly identified by Mona was GPA82V. So as you can see, it describes a position in the GP. So who remembers what GP stands for? Does anyone remember what GP, how do we interpret this specific mutation? Where is it located in the genome? Okay, well, let me remind you, GP stands for glycoprotein. So we'll talk a lot more about glycoproteins today. And this annotation A82V stands for position 82. It's an amino acid position. And the change happened from A in the reference genome and V in the specific strain that we're talking about. And so one of the things that they discovered was that potentially this specific mutation is associated with the viral entry. And so we'll talk a lot more about viral entry today. Others that posted on the forum have posted some responses to the previous assignments. So thank you, Anita, for sharing your results here, and also some technical challenges. And so today, we'll try to address some of these things. Briefly, for Safa, I saw that you did not have a chance to get results from your analysis pipeline. And so to explain the reason for that, I just want to make sure that the names of files are going to be sensitive. So try and not include spaces, underscores, and all kinds of characters like that in your input data. Make sure that you're matching your data with the right kind of a reference. And so I think for Safa, for your issue, Harpreet is posting a response to the issue on the forum. But to summarize, that is the issue. The issue was that the name of your input file did not have or had this special character. And so because of that special character, you were not able to get the results. Did anyone have any other technical questions about the paper that we reviewed on the glycoprotein and the specific mutation and how it is associated with the disease phenotype, or any issues with the technical pipelines that we've run up until now? Okay, so if there are no other technical questions, I hope that most of you will be able to follow the directions posted on the forum. If you have any questions or technical issues, please do let us know. We'll be looking into those. We have here Harpreet, who is experienced with looking through the different technical aspects, and she'll be able to help you with those. And also, hopefully, you're finding all the data, so you should be able by now to do multiple sequence alignment between different types of sequences that you have selected based on a scientific question or a research question that you might have, to be able to determine the specific differences. And hopefully this particular publication helped you kind of see how others have done it, and how you can do the same. Now, I did want to mention that we are going to have a program coming up called OmicsLogic Transcriptomics, and we'll have a free webinar. In a couple of sessions from now, we'll talk about the host response. And as we speak about the host response, we'll be talking about how to measure levels of gene expression, annotation of pathway analysis, and how to interpret the different profiles in the gene expression on bulk RNA and single cell RNA levels in this upcoming program. Now, it starts in December, but I did want to mention to everyone that this is something that if you're interested to continue building up your bioinformatics skill sets, it's a program that will be hosted in December, but we will start with a free webinar. So you're welcome to join that program if you are interested. And we will be following up by email with the details on how to register for that. So let's review what we covered in our last session. In our last session, we talked about how a single disease caused by a pathogen in an individual host might spread rapidly, which is called an outbreak, and how those specific diseases that cause outbreaks in an epidemic or a pandemic are typically associated with zoonotic transmission, and how we can incorporate that into our analysis, into the bioinformatics component. And so when we talked about this session, we talked about two different types of approaches that you could take. One, to ask the question about where did the virus come from? So basically that boils down to zoonotic transmission, geographic location, and it helps us identify a novel strain of a virus that could be different from established strains. So for example, with coronaviruses, there are many coronaviruses, many of them are mild. The one that is causing the current pandemic is different from the ones that we know and have been studied extensively. And so one of the first questions is to identify how is it different and where did it come from? So what is it? So it's really to define what it is. And then the second type of a question that we can ask based on sequencing data is where is it going, or how does it work? And what are the specific biological differences that result in the phenotypic differences that we can observe? For example, those could be rates of transmission, mortality, speed of disease progression, etc. And so the follow-up questions that we can start asking based on our understanding of the biology or the phenotype are related to intervention. In the case of Ebola, we reviewed the publication that describes how specific locations where the Ebola outbreak happened or has been happening, these outbreaks have been happening over the years, have been linked to these natural habitats of the hosts where this Ebola virus might come from. And we talked about some of the differences in the immune systems of these different hosts, like bats and the intermediate hosts, like primates, and the host of the humans that eventually where this pandemic has spread so fast. And so there we talked about how we can make those comparisons, and sequencing data allows us to identify the strain that we sample as a close relative of a strain that we can find in the host animal, for example, and that helps us establish that that is the primary source of that infection. It's an important consideration because of the interaction. So prevention of such outbreaks, containment of those outbreaks has to do with a clear understanding of epidemiological factors of where the disease originated. We also talked about how then the second type of a question, right, what we already kind of briefly discussed, was to understand how, as the disease spreads through the human population and it evolves, and you can see this phylogenetic tree that represents the evolution or the separation from the original strain into multiple additional strains, how that could be broken up into lineages. And those lineages could be associated with specific types of changes at the amino acid level, ultimately biologically being interpretable from the perspective of understanding what each protein does and how those specific changes could result in a different function. And that function, in this case, was linked to this glycoprotein, as we saw before. So the glycoprotein was subsequently an important factor to design vaccines and antiviral treatments that would prevent this virus from infecting whole populations. So we talked about the glycoprotein, and we talked about how to find the glycoprotein or any protein that has a known three-dimensional structure on the protein data bank. And so this is a review of the different types of proteins that the Ebola virus has. And we can see here how the virion, or the actual virus covered in this membrane with these glycoproteins, how it's organized. And the glycoprotein here, we can start mapping those mutations onto the glycoprotein structure. So in terms of this assignment, the objective of this assignment was really to help us understand how to read through a publication, identify the specific data sets which were used to answer a question, and then how to make sense of the analysis results, right? So how to analyze data, how to have a specific analysis design constructed, right? So when you have a question and you have a data set, how to translate the question into an approach to data analysis. And from the data analysis results, how to interpret the analysis results so that you understand the link between what was found in the data and how that addresses the question at hand. First of all, I know that Mona answered the question. I don't know, has anyone else attempted at reading through this publication and understanding what's going on there? Okay, thank you, Babis. And what were your conclusions? What did it clarify how they use data and bioinformatics analysis that helps address this question about, why is this Ebola virus spreading at different rates in different locations, and how we can understand that phenotypic difference with how can we link it with genomic differences. Okay. Yes. Yeah, it is a long answer. I mean, you're welcome to unmute yourself. I just want to make sure that we, maybe for the benefit of also those that have not had the time to read, we can briefly discuss this project. Okay. Yeah, if you would like to just comment on this publication, if you had the time to read it.
Hello?
Yes, hello, Babis.
Hello. Yeah, it was too long to write it to answer all this in the chat. Yeah, it's a very interesting paper. It discusses a lot about how the virus spread and why there were different rates of spread in different areas. There is not, I mean, from what I understood so far, there was not a single reason. There were multiple reasons for that. Of course, they talk about a lot. They talk about the glycoprotein and how changes in the glycoprotein, which is like this, what they call like a spike, like it's the protein that attaches on the host cell and makes the genetic material of the virus to enter the cell of the host. Different mutations there will make the virus be more virulent, so it's easier to infect someone, or less virulent. That's one of the arguments made there. There was another argument that had to do with the fact that Ebola virus is not like the natural host of Ebola virus is not human. It's the bats. So only humans are only circumstantial hosts for this virus. And this is the reason why it's so deadly. The virus has so high mortality for humans because the virus doesn't have this evolutionary pressure from to because the viruses generally, they argue that the virus generally um, when they don't kill the hosts, their hosts, they are in a better position because they can sometimes infect more people, the infected host. But if they kill the host quickly, then the virus will die in a way with the host. But this doesn't apply to Ebola virus because the host, the natural host for Ebola virus is the bats, and they actually don't infect the bat. I mean, the bats don't die from the Ebola virus; they can handle it, but humans cannot. So depending on what strain from what bat start infected each area, they expect that the widespread of each viral strain can be different. So basically, there's one perspective, evolutionary perspective, and the other one is a more molecular perspective, as I said, about that has to do with the glycoprotein, the spike of the virus, and the way it can attach to the cells and infect them. I hope I covered it. I don't know.
Yeah, thank you. So it's a great summary. And I think that obviously many reasons for why the virus spreads include non-genomic reasons for different types of healthcare systems and different types of prevention strategies and other issues as well. But one of the key things that they focused on were the differences between these lineages and the apparent correlation between that mutation appearing common to this particular group of genomic sequences. And given the number of sequences that they had, it's a good assumption that it contributed or was somehow associated with that particular strain that spread fairly fast. So thank you for that great summary, Babis. I also noticed that similar types of analyses could be found in other publications. So this was just one of the publications. If you were looking out into other research studies of the Ebola virus epidemic, you probably noticed that the most recent ones are the about the outbreak that happened just more recently in 2019, I believe. And one of the questions that you probably noticed is that they were trying to understand, is it the exact same virus or did it somehow change? And so I think that's a very important trend that we can take away from that Ebola virus, an epidemic that happened over the last few decades. Remember, the first one that we looked up was in the 1970s. We have data from the 1970s all the way up until 2019, so very recent. And we can therefore ask very specific questions that could be answered fairly definitively with the type of analysis that we're speaking. A similar situation, a little bit different, but very similar, is something that we can talk about briefly today as we'll focus on the glycoprotein from the coronavirus. So one of the first questions, as you all probably remember, in early 2020 or the end of 2019, was the question of whether the virus was engineered or whether it also came from a similar situation where it persists in a host and could come through an intermediate host in close proximity to humans and then spread very actively, having higher rates of mortality. And therefore, we can kind of clarify whether it's a naturally occurring phenomenon or was it potentially engineered in the lab. And so in May of 2020, or maybe in March 17th in 2020, there was this article that we can kind of use as an introduction to what we'll do today. And this started from a letter to the editor. It was called The Proximal Origin of SARS-CoV-2 and was kind of a logical approach to try to understand where did the SARS-CoV-2 virus strain come from and what are the specific differences in this genome compared to other ones that are found in some other common reservoirs for the coronavirus. And those are primarily also bats. And so here you can see a comparison of human SARS-CoV-2, bat, pangolin, and some previous SARS-CoV, so SARS-CoV-1, and some other bat SARS-CoV related. So for example, the reason for this approach, the logic behind this approach is that SARS-1 and also MERS have thought to be originated in other animal hosts, for example, camels in the case of MERS, Middle East coronavirus, and SARS-1 was also linked to bats. And so those were known and well studied and well understood in terms of their ability to infect specific types of tissues, cause a specific type of a lung disease. And so enough information was there to try to understand what is going on. With this novel coronavirus, everybody was trying to really understand the differences of this novel coronavirus from those more understood viruses. And so here, what they did in this publication is they took these sequences, analyzed their genomic differences and specific regions in those differences, where those differences have been more pronounced, have been identified. So you can see here, this is the full genome, and this full genome contains multiple protein coding regions, and those include this specific region for the spike glycoprotein. The spike glycoprotein has multiple functional domains. We'll talk about those in greater detail in a little bit, but those functional domains have these very specific differences. So what do we mean by very specific differences? Two things. The first one are the common differences, right? So here you see the common, that's in A. And here are the different, the B. So what are they? In A, we see a common difference that is common for the SARS-CoV-2, RADG13, which is also a novel genome from one of the bat, and pangolin. So these are common between the three. And then they are different from these bottom three, which are all of the SARS-1 related. So human SARS-1 and older bat sequences for SARS-CoV-1. So that gives us a sense that if they are common between these three, but different from the older three, that might be an important change that we can consider in this framework of logically establishing what is related from where to where. Here you can also see that there are some closer relationships between the pangolin and the human as compared to the human versus the bat rat G13, right? So here you can see, for example, these two are the same, these two are the same, and these are the same. And so these are found in a specific functional domain called receptor binding domain ACE2 contact residues. So because we know how the glycoproteins work, and we'll talk about that in a second, we can start putting together these pieces of information and making sense of them. Let's take a look at some of these different ones. So these are the unique sequences to the human SARS-CoV-2. So they are not found in any of these similar viruses that infect bats, pangolins, or previous SARS-1. And so the second question that you can ask is, what biological differences are we to expect from these unique changes in this novel SARS-CoV-2 genome? So how do we start answering that question? So let's think from the Ebola virus example that we already did, and let's apply the same ideas to what we can do with this SARS-CoV-2. So the first thing that we can do is go to NCBI virus, and we can find all of the same FASTA files that are, the data that we have. So I don't know if many of you have looked, but all of these sequences are present, right? So just so that we will refer to this in our future sessions, let's try to do this together. So let's go to NCBI virus. So on NCBI virus, just so that we can replicate the exact same analysis as was done in this paper, we can look at the SARS-CoV-2 sequences, right? So we can go search by virus, and let's put in corona. Okay, coronavirus. And remember, what did they compare? So they compared sequences from the novel coronavirus, SARS-CoV-2. They compared it with this RADG13, and then they also had one from pangolin, right? So those are three different hosts: human, bat, and pangolin. So we can go into the host right here and select human. We can also add pangolin. So these are the pangolins. Well, these are all of the pangolins. And bat. Okay, so here we have three hosts, and then we want to find specific types of sequences. So for example, here we can see, all of these are in different locations, obviously. We want to find the ones in China, right? Because geographically, that is where they would be originating from. So let's find here Asia, China. Okay, so we know that geographically they originated in China. We want to compare in China at least 3. So the human SARS-CoV-2, bat SARS-CoV-2, bat SARS-CoV-1, and then also this pangolin. So let's see. So here we have, again, we have a lot of ref-seqs, and those are good because those are the commonly used references. So we can look at the dates here. And here we see this is a human from SARS-CoV-2 because it's in 2020. So we can select this one. This is from 2016, right? So they're kind of organized here by date. So here we can see that in terms of the ref-seq, we can take as a reference one of the older ones. So for example, here is the bat coronavirus HKU10. And here we have these different types of other alpha and beta coronaviruses, right? So just so that we can go through this, let's take this HKU. So one of these, we know that this is a much older one. We know that these are very different from the SARS-CoV-2 sequences. So we'll just keep those. Okay, here you can also see bat coronavirus. So these are now 2020s, right? And one of the things that we need to find is a sequence from pangolins, right? So if we go through here, and let's keep now, let's download these. Well, let's in fact maybe download them separately. So I'm going to take where did we go? Okay, so yeah, so this is the human one. I'm going to take that one, and just so that we can have another point of reference, let's take another one. So remember that this is also the length, and the length is important here. We only want to take the ones that have a full length. So let's try to go through here and nucleotide completeness. Let's just select the complete ones. Okay. So once we select these complete ones, again, I'll select this one back. We can find, okay, so here's another one from 2020. Let's see, what else do we have here? Homo sapiens. Okay, so here we have a pangolin. So let's take a pangolin one. And here we have another one, a more recent one from a bat. Okay, and here we have another number of different coronaviruses. Not all of them are related to SARS-CoV-2 and others. So let's try and find, okay, so here's severe acute respiratory, so this is from 2017. So let's select these also. Again, these are the older human coronaviruses. Okay, so here we have several sequences that we've selected. We can download them. Nucleotide sequences, download selected records, and click on download. Okay, so in our downloaded sequences, let's just take a look at what do we have. So remember, we can look for this symbol to give us all of the sequences. So this is from Wuhan, HU1, complete genome. So this is the first one that we have. So 1, 2, 1, 2. Okay, so here's one SARS-CoV-2 from the human. Here's another one, pangolin, right? And here, bat coronavirus RAG-G13. Remember, they had that one as well. So we have a lot of these similar sequences to what they did, right? So if we spend a little bit more time, we can probably find better ones, but for now, this is fine, right? So we have 6 different sequences here. Let's now go to the platform. Again, I'm not going to pause here too much because we want to make sure that we do everything that we have planned for today, but I do want to just go through this practice one more time because we'll discuss much more in detail how to practically do all of this in our next session. But here we'll go to the phylogenetic tree, okay? And then here we will select FASTA files. We'll then upload these files. And I'm just going to change the name. Well, so Anita, I'm not going to share with you the file. I will in our next session, but in this one, part of the experience, I think, is being able to find those and read through the different annotations. So that will be kind of your assignment to try and do this as we do it together. If you didn't have a chance to do the same thing, please try to find them on your own. I think it's an important part of translating your understanding into a practical skill of being able to find those data sets. So I'm just going to call this SARS human bat pangolin. Okay, so here I'm going to find it. Okay, it's uploaded. We don't have any groups, so we'll just put in group A. Now we need this GenBank file for framing codons. Where do we find it? Again, I'm not going to give it to you. You can find it on NCBI, and you can follow along with me to try and make sure that you can find it on your own. So again, here you can see that we can look for SARS-CoV-2. And here we have the reference genome sequence, right? So this reference genome sequence is what we want. We want to go to the RefSeq genome. And remember, in here, it tells us what we need. We need a GenBank file, right? So what is the GenBank file? The GenBank file is the one that contains the translation of the nucleotide sequence into the amino acid sequence. And that gives us the framing of codons. So here we want to do a file and select right here GenBank. Create that file. Again, we'll do all of this again next time. Please try to do this on your own first. Okay, and then let's upload it right here. Once you have done this, in this case, we can also add an older and a newer genome just to kind of give us that reference, but we're going to skip it this time. We're not so interested in the timing. We're more interested in the origin. So now let's do start. Multiple sequence alignment. Convert it to amino acid sequence. Select the different codons. We don't have to do anything here. And then here, let's just do all phylogeny methods, nucleotide level, and then tree space, and then end. Okay, so if you have not been following along, you don't have the data, please try to find the data, run this pipeline on your own. It's okay that your data is a little bit different from what I have. It's actually fine. You can then in our next session, try to interpret the differences that you find. Okay, so I'm just going to call this SARS-CoV-2 origin study and run pipeline. Okay, just to get a sense of where everyone is, how many of you were able to start this pipeline or a similar one to what I just did? Was anyone able to start this pipeline that I just did or a similar one? And Anita, sorry that I'm not giving the file, but have you been able to find those sequences on NCBI virus? Okay, I'll take that as a, okay, yeah. So Anita, again, don't try to replicate exactly what I did. Try to follow the logic of this analysis. So the logic of this analysis, again, remember, it's based on this publication. You can see in this publication what they did, right? So you're looking for sequences that are from human SARS-CoV-2, the bat similar to SARS-CoV-2. So you know that in terms of years, it's going to be recent. So in 2020 or end of 2019. Pangolin, again, you want it from year that is very recent. So 2020 or the end of 2019. And then these are some of the older comparisons, right? So this is kind of like your negative control, so to say, right? So these are human SARS-CoV-1 and then bat SARS-CoV-1. So these are going to be called SARS-CoV related. Okay, and don't try to replicate exactly what I did. Do your own understanding, your own interpretation of the objective here to try and just perform the alignment. Okay, was anyone else able to run the same pipeline or prepare the same pipeline? Okay, so I see that no one else has really done this yet, but hopefully you can review what we did today in a video, try to do the same steps. Again, it should be by this time fairly straightforward to go through all of the available sequences, select Coronaviridae. Here you can select more specifically the types of viruses that you're looking for. You can also make different groups. So you can download several different FASTA files from here on your own. You want to look from China and then different hosts I have right here. So you're welcome to try and do the same thing. So this is step number one, right? So step number one, we found those sequences and we downloaded the FASTA files. Then we went to NCBI RefSeq and we downloaded this genome as a GB file in GB format. So that format gives us the translation of nucleotides to amino acids. Then we went to the server platform and we can either use the phylogenetic analysis or the quasi-species section, and we can upload the data there. In this case, you can see an example. We can also separate it into different groups. If I download several different groups of, so several different files, I can separate them into groups. And then we can build a pipeline. So the key to remember about the pipeline is not exactly how it looks, but what is the logic? And the logic here is start, multiple sequence alignment, and then all phylogeny methods in tree space, and then end, right? So we're just going to try and find differences and study those differences in terms of protein sequences. So this is one example. This is based on different groups. And this is another example. This is based on the specific types of sequences similar to what I just did. So here we have COVID-19. So this is the human COVID-19, bat coronavirus rat G13. This is a coronavirus genome found in bats around the end of 2019 or early 2020. And then these are some of the older ones. So here you can see SARS coronavirus SARS coronavirus or VANI. So these are human ones, and these are bat ones. So the differences in terms of their relationship is that COVID-19 is the closest to the bat coronavirus rat G13 and more distant from the SARS-1. And the second thing, as we did last time, is remember we talked about this TSV file, the file that organizes the sequences into their nucleotide and translation into amino acids, where we can study similarities and differences between those. And we'll do this in greater detail in our next session. But the important part of it for today is that we have a similarity plot. This similarity plot shows for different types of viruses. So here you can see several different types. Pangolin is blue. Yellow is COVID-19. This is a reference. So you can see that the reference COVID-19 is almost identical to the reference, which was COVID-19. And bat, right? So the question that we want to ask here is whether pangolin or bat is closer to the human. So you have a pink one, which is bat, blue one is pangolin. Please tell me, looking at this graph, what are some regions that you can identify on this chart that show a dissimilarity between bat and pangolin? Okay, so obviously there's dissimilarity between bat, pangolin, and human. But now the question is also, where do the bat and the pangolin sequences differ substantially? Does anyone see any regions where those two are different substantially?
Uh, yeah, Ilya, it's almost in the middle where the pink one is going above and the blue one is going down.
Right
So
here.
one is there, yeah, this one, and few a little bit ahead, where also like above this, you can see the average. There's written average over above, just above it, the average. There's also some differences over there on the left hand side of your cursor.
Here?
Left and no, no, not this side, just
Oh,
opposite. Yeah, this one, this just near, yeah, this one.
yep. 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 So you're right. So a good way to understand the difference is to study the differences below or above this average line, right? So we can take the average line and we can say, okay, so if we have a position where the red is in between separating the blue and the pink, we know that they're substantially different. And so if we kind of look across, correctly identified here, we see here's an example, here's an example, here's another example, here's another example, right? So these are special regions. It's not that it is across the whole genome that we see a very similar difference. So if we combine two measurements, one measurement is where both are different from human, right? That's question number one. And question number two is where bat and pangolin are different from each other. So overall, we see a dip here, but this dip seems to be very similar between bat and pangolin. And here we see another dip. And in this dip, there is a pretty big difference, right? So those differences, for example, they are still around the 80% range. These go below, right? So here we have this going below the 70% range. And so this becomes an interesting region for us to focus on. If we compare this to the sequences of proteins, we'll see that this specific region falls directly in the spike glycoprotein. However, not equally. We kind of see that there are two sections here, right? So there's a section that has a lot of dissimilarities, and here's a section that has somewhat close similarity between them. And so this kind of an annotation helps us convert the genomic analysis and start thinking about specific proteins and what might the changes indicate from a biological perspective. Now, before we continue, I do want to mention that there are several packages and tools that you can find that do this kind of analysis. This is a package in, I believe, like a Windows type of a Windows program that you can build a similarity plot similar to what we just did and study the similarity. There's a package in R that looks at similarity and difference in specific domains and different types of sequences. And we'll talk about this as well. And here's also another package that essentially builds the same thing. The way that I produce this plot is in Excel. So based on that TSV table, you can produce this plot in Excel, and we'll do that in our next session. So in our next session, we'll do a practical hands-on based on this kind of an analysis. So here you have your assignment for next time. This is the assignment for next time. So this is something that will help you prepare for our next session, because we're going to be talking about the protein analysis. Okay, so today what we'll do is we'll prepare the setting for converting this kind of an analysis of similarity into the specific differences between proteins. Okay, and that's what we'll do today. So today we'll talk about disease progression and outcomes, and we'll talk a lot about glycoproteins. So what are these glycoproteins? The glycoproteins are one of the major components of human pathogenic viruses, but they're also used in human cells. They're also used in bacteria. So they're really just a whole category of proteins. But specifically in viruses, they have been demonstrated to have important roles in infection and immunity. So here you can see the HIV virus, and this is a fully assembled infectious virus, which is known as a virion. So it's a particle. The simplest virions consist of two basic components, namely the nucleic acid and a capsid, which is a protein coat, functions as a shell to protect the viral genome from the nucleases. So we have inside the genetic material, and it is covered in this protective membrane. The capsid comes into play during infection to attach the virion to specific receptors exposed on the prospective host cell. The capsid proteins are coded by the viral genome. Due to its limited size, the genome codes for only a few structural proteins, and capsid are formed as single or double protein shells and consist of only one or a few structural protein species. So here you can see this is the HIV glycoprotein, and this is what it looks like. This is the virus membrane. This is the human cell membrane. This is kind of how it attaches to the human cell membrane. Here you can see the dengue virus. So you can see this pattern on the dengue virus, which essentially shows how many of the same protein structures are packaged together to form this coating. And this specifically, you see the little red positions here, these are these glycoproteins. In SARS-CoV-2, the same thing. We also have this kind of a structure that goes into the membrane. This is a lipid, and here you can see the glycoprotein sticking out. These multiple protein copies must self-assemble to form the continuous three-dimensional capsid structure. The structural virion proteins are extremely important to the virus so as to facilitate the transfer of viral nucleic acid from one host cell to another. Glycans are major components of the outermost surface of viruses. The majority of infections of viral pathogens with their host are influenced by the pattern of glycans and glycan binding receptors that each expresses. And glycans are the most complex biomolecules due to extensive branching of carbohydrates, and a variety of glycoproteins have been identified that are specific to different types of viruses. And these pathogenic glycans are either virus encoded or host derived, usually, and they elicit specific humoral responses in the human body. So just that we see the same protruding structure on the surface does not mean that they act exactly the same way. They interact with different types of receptors on the host cells. For example, flu, influenza virus interacts with sialic acid. We'll talk today a little bit about furin that acts as a activator of the SARS-CoV-2 particle. The HIV glycoprotein interacts with the T cells. So we have different types of glycoproteins. Their structure is typically that they're exposed on the surface. They're made to interact. Let's talk about what are some of the forces that cause these particles or proteins to interact with different proteins. Now, if you think about this, this is the structure or the sizes of these particles. You can see here the smallest shown is the Zika virus. Here you can see a bacteriophage, coronavirus, fairly big. And then you can see bacteria, right? So this is the scale, all the way to a hair follicle, right? So this is a long hair, but this is kind of like an individual piece of it. Now, what type of a force do we know that causes hair to interact with things? One of the things that you can think of is static electricity, right? So static electricity causes this whole hair follicle to stand up, and if you have a balloon or something like that, right, it will attach to that. So similar forces actually have a huge impact on these different small, tiny little particles. So two things we need to remember. The first one is this electric capability to move and attach between different particles. The second one is pH, right? So all of us know different pH scales. We have more acidic, less acidic environments, and those have a profound effect on different types of substances. So these two forces are going to be very important when we talk about protein structures and how the specific changes in those protein structures affect their ability to bind and interact with other types of proteins. In the case of this novel coronavirus, or coronaviruses in general, the way that they infect cells is by attaching this glycoprotein to a receptor. And then this is called the process of endocytosis. In other words, the virus comes inside the membrane, and the objective is to reproduce based on the genomic information that it carries inside. So this membrane needs to be disintegrated, and the inside material has to unfold. So if you think of that force of the static electricity, or the way that pH affects different types of structures that maybe out in the air are very solid, right? If you think about a pill going into a high pH environment, it will disintegrate. That is how we think of drugs, right? Drugs could come in tablets. Inside they disintegrate and the individual molecules spread through the body. So similarly to the viruses, that's what happens. It's hard surface on the outside, or maybe more like a lipid, and then goes into a different pH level and starts changing in its shape, interacting first of all with the receptor, and then being able to extract this genomic material into the cell. So inside the cell, first of all, the ribosome produces a positive sense of this RNA, because it's an RNA virus. And then the viral polymerase actually starts replicating the pieces of information that are needed to produce the specific proteins that will eventually exit the cell and form virions. Now, the key moment in this graph is to understand the function of this glycoprotein. So this is the SARS-CoV-2 virion. You can see here a more visually accurate and appealing kind of visualization. You can see these, the virion itself has these protruding glycoproteins, and the glycoproteins contain multiple functional domains. So what are these domains? The overall protruding part of the virion is called the ectodomain, so it's outside of the membrane, and it separates into the S1 and the S2 subunits. This whole protein consists of 3 chains that are folded together to form this particular protein. So when we study the nucleic acid sequence or the nucleotide sequence of this protein, we can start thinking about interpreting how the specific changes that occur in several different ways. So what are these ways? Protein function could be determined by its sequence similarity. If we have other sequences that are well understood and studied, we can compare sequences. We can also think of 3D structures, and 3D structures fold. So we'll look at how do they form actual protein 3D structures and what are some of the moving parts of those structures. Or we also in the future will talk about high throughput data that could be generated to explain what proteins interact with and what molecular pathways we could analyze for functional impact. So this is kind of like a framework for the analysis of data and linking between the genomic data and the 3D protein interaction level. Now, in this case, we have this alignment, right? So I'm just giving you here an example. After our analysis finishes, we'll take a look at what we have. But right here, we have this comparison based on COVID-19, right? So this is human COVID-19, SARS-1, which is also human, bat SARS-1, pangolin, and MERS. And you can see at the level of amino acids, right? So these are amino acids, not nucleotides. We see specific areas which are very different, right? So those are the areas that we want to focus on, but we don't want to just understand it at the nucleotide level or amino acid level, the sequence level. We want to think about proteins. A key component here is having a reference model, right? And so here's a model. It is stored, we talked about this last time, on the PDB bank, data bank called Protein Data Bank. And let's take a look at this specific structure. So this is the specific structure here. This is the structure that you would find on the protein data bank. So let's go and download this protein model. How many of you already have UCSF Chimera installed? Okay, so for those of you that already have Chimera, we will download this structure and open it in Chimera. For those of you that don't have it yet, please install it. Okay, so here is where we can find the specific model, 6VXX. You can see it's the structure of the SARS-CoV-2 spike glycoprotein in the closed state. So we'll talk about these states in a second. But here we have these closed states. And let's go to UCSF Chimera. Okay, so how do we get from having this link to opening it in Chimera? First of all, to download UCSF Chimera, this is a link for those of you that have not downloaded it yet. You will find here, this is Chimera X. So there's also an older version called just Chimera without the X. But here you will see some of these are very simple to download and install. And once you install this, you will see something that looks like this. Okay, so here Let's close this session. Okay, now we want to download this. So here you can do open. So this is the simplest way, is you can just open this from your computer. So once you download that file on your computer, you can do this. And we'll talk about how to actually download directly from PDB in our next time. But right now, what you want to do is you want to just download this model onto your computer and then open it in your Chimera. Okay, so does anyone apart from Manav have Chimera already installed? Okay, so for those of you that have this, Manav, were you able to load this model here? Okay, and I understand that nobody else still has that, so please, for our next session, make sure to install. Briefly, let's take a look at this model. So once you open a model, it will come up here on the side with some information about what we just did. So for example, if you want to just do it without downloading, you can just use this command line to call different commands and open or change the way that this model looks. So here we have the 6VXX model. And you can see that it has 3 different chains, right? So we can look at one chain. For example, I can select this chain. Here you can go to molecule display. So here I can just hide everything. So I'm just going to hide this and also hide this. So here you can also see we have 3 different chains. We don't want to see the missing structures. Okay, so this is one of the chains that we have. And here we also have the non-standard residues. For now, we can hide them. So this is just one of the chains that we have. Okay, so here we have one of the chains, and we can look at this chain as a surface. Okay, so this is the surface. It looks like a needle that is folded together. So it looks like a knife. You can see that it could unfold, right? So this piece could go down, this stays up, and it becomes a functional piece that the virus can use. And we'll talk about this process in our next session. Here we can see how these 3 different chains, right? So if we go back and we select this one, we can also show that piece, right? They are assembled together. Let's also select the A and show it. Okay, and here we see how they together, the 3 chains, form this one protein. Okay, and this protein, remember, has functional domains. So let's see how we can find the association between sequence and this 3D structure. So here you have this little icon that shows the sequence. And here we're just showing chain A. They're exact replicas, all 3 chains, so we don't have to worry about all of them. But here is our sequence of amino acids, right? So for example, I can select a certain portion, and then I can see where that sequence is, right? So here's what I selected right here. Okay, so here I've selected this, and it's right there, right? So now I can find how the specific portions of my sequence are mapped onto the three-dimensional structure. And we can look at it from a number of different ways to visualize. We can look at it from the perspective of a surface, or we can look at the perspective uh, of a ribbon. And the ribbon gives us a better information about the folding of these amino acid chains, right? So remember what we said here is that it's actually a string of amino acids that folds together to produce this functional piece. Okay, so here, once we know what region we are looking for, and again, let's take a look at this region. We have this region right here, right? And it is the letters NSPRRAR, right? NSPRRAR. So how would I find this right here? I would need to know the position, so where it starts from, and look for that sequence, NSPRR, right? So if you hover over the individual letters, it will show you what is the letter and what is its position. So remember here, we have the positions in nucleotides. So these are nucleotide positions, and we are looking at amino acids. And those amino acids, in terms of its ultimate position on the full genome, is going to be very different from the position specifically on the protein, right? So in the protein, now we have to start thinking about the specific types of sequences that we need to consider as we start mapping out the differences in nucleotides all the way to their structural implication. And by the way, those of you that are very new to Chimera, here's a sequence of videos that I recommend watching so that you can start learning about some of the basic functionality in terms of locating the sequences and interpreting them from a surface and interaction standpoint. So what are these functional domains that we'll talk about in terms of this glycoprotein? The glycoprotein is encoded in this S gene. This is the gene sequence. And the specific domains are mapped onto the sequence in the following way. We have the S1 and the S2 components. And then within the S1, there are the signal peptide, receptor binding domain. And in the S2, there's a fusion peptide, HR repeat, heptad repeats, transmembrane domain, and the cytoplasm domain. If we look at the S1 subunit and the S2 subunit in terms of this glycoprotein, we can actually see them as a cap on top of a needle. So you can see that the needle kind of goes inside. You can see the red right here. And here you have this cap sitting on top. The way that this protein becomes activated is that it is cleaved in the connection between the cap and the needle by furin. Now, furin is a specific type of an enzyme which causes proteolytic cleavage, which actually regulates numerous processes in different aspects of health and disease. It's a ubiquitously expressed serine protease. In other words, it's expressed in many places, many tissues and places in the body. And it cleaves a plethora of proteins at this polybasic recognition motif, which is a specific sequence of amino acids that it tends to recognize. Mammalian substrates of furin include cytokines, hormones, growth factors, and receptors. So it's not surprising that aberrant furin activity is associated with a variety of disorders that include cancer and many other diseases. The enzymatic activity of furin is also exploited by numerous viral and bacterial pathogens, which enhances their virulence and spread. So one of the key portions of this activity in terms of SARS-CoV-2 is that this specific region, it's important for the pathogenesis of this SARS-CoV-2 COVID-19 disease. and it causes cleavage in these polybasic regions. Once they are cleaved, so they are kind of like these exposed regions, once they are cleaved, you have this top cap that falls off, essentially. Now, what is this cap a part of? Do you remember? Is it a part of the S1 subunit or the S2 subunit? Who remembers which part is the cap? Yeah, does anyone remember? S2, it says Anita. Anyone else? Okay, so the truth is it's S1. So the S1 subunit is the top one, right? It's easy to remember the S1 is the top one, S2 is the bottom one. So the S2 subunit contains an important functional domain, which is called the receptor binding domain. This receptor binding domain is important because this specific portion of this protein is what binds to the receptor. Since it binds to the receptor, the recognition or the binding domain, the recognition between the receptor and this protein is a critical area where we expect a lot of variation if we talk about different hosts. If we talk about bats, pangolins, and humans, they have different receptor structures and surfaces. And because of that, when the virus goes between one host to the next, it has to adapt. It needs to change that region. Now, if you look through the literature, you can see that the top portion, right? So here you can see it is more variable than the bottom portion. Why is that? Well, because if it goes between different hosts and needs to adapt to different hosts, that changes. But the way it functions, the main functional part on the bottom, the S2 domain, is still acting in the same way. It performs the same function. So it is more conserved because that function is critical to not change from the perspective of the virus entry. So how does this whole thing work? Here's a little animation. So this top S2 subunit, you can see it contains this receptor binding domain, which is not static. It actually changes. It goes up so that it can connect to the receptor binding domain. And because of that, this is an important portion. So we can actually map this. You remember I said that this is a closed state, the model that we have. We can also find the open state. And what we saw in this animation is kind of a transition between the closed and the open state. And so here's how it works with the ACE2 receptor. The ACE2 receptor, this is just a portion of the ACE2 receptor. It comes and connects to this receptor binding domain when it is in its up state. And here you can see that the receptor binding domain, the ACE2, are connected. So this is a critical part for the recognition. And there, obviously, we should expect a lot of differences between the human, the pangolin, and the bat coronaviruses. Some of the other important functional domains in the S2 subunit, the bottom part, are the heptad repeats, so 1 and 2. You can see the heptad 1, the heptad 2, and then the fusion protein. So these important portions are more functionally important to the virus itself. How do they actually work? Here you can see that the proteins that attach to the receptor binding happens. There's the proteolytic activation, so this is cleaved off. And then you have the bottom part, this functional part up here. And then what it does is it transforms, because this cap is kind of taken off, it transforms and it becomes a fusion of the two membranes, which are bridged together, essentially opening a hole in the cell membrane. And through that hole, the virus can inject its genomic material. Okay, was this somewhat clear in terms of how we got from the sequence to the protein and what is its function? Does anyone have any questions about this? Okay, to kind of understand more about this, please take a moment to read through the publication in this link. Okay, and that will give you a much better understanding of what this virus does with this protein. It's called the structure, function, and antigenicity of the SARS-CoV-2 spike glycoprotein, which describes how it enters the cell and what are some of the general functions as well as some of the components that we're going to be talking about. Now, if you already have, how did we find what all the functional domains? Well, these are the types of studies that are described here. And so in the study, you'll see what the different functional domains are. How can you understand what are the different functions of this protein? Here are some methods. So one method is you can use amino acid sequences of those proteins, and you can compare them to other known proteins to identify motifs, which are specific patterns in the sequence, or whole regions that are identical to others. So for example, as we said, the SARS-1 and MERS viral sequences have been around since 2002, 2003. And so people have done a lot of experiments by identifying what are the different functions of the different proteins and their components. So now we have the SARS-CoV-2. We can go back, compare the sequences, and say, oh, okay, here's a sequence that's identical. It probably does the same thing. Another is we can actually look at the 3D structure. If I find something that looks like a knife, I think it probably acts like a knife. If I find something that looks like a ball, I'm going to say probably works like a ball, right? So by looking at the 3D structure and similarities to other things that we know how they work, we can look at them. 3D structures have functional properties. Remember, we talked about electricity, pH, and we can start thinking about the interaction between different types of materials and environments. Electricity binds different atoms together. Hydrophobic and hydrophilic surfaces help explain where they're positioned in terms of membranes and which ones are facing outwards. And so we can start piecing together that information. That information is known based on the different properties, physicochemical properties of amino acids. And then finally, how we can study them is we can develop different types of variants. So we can introduce mutations, cut out whole sequences, or insert new sequences and see what would be the result. And so those are some of the ways where we can study the individual proteins and their components. Does that answer your question, Dr. Jang? Okay. Okay, so for our next session, please take a moment or please take some time to read through that article, download and install UCSF Chimera, and try to find in this sequence some of those regions that we will find through analysis. So in the analysis stage, if you have the time to practice your analysis, we've looked today at comparative analysis of genomic sequences from multiple hosts, pangolins, humans, and bats. Based on that alignment, we will find some differences in the SARS-CoV-2 sequence of the protein. We can identify those sequences in this in this 3D structure. And then we can try to compare them and think what might be the implications. So Anita is asking, can we make our own 3D structure from the sequence if they are not available? It is possible, but yes, through homology modeling, which means that you have some reference and then you have some variation and you can kind of predict what might be the change. It's not an exact science in terms of many biological considerations around the folding are not going to be necessarily 100% accurate. You can see that example by looking at this little animation, which was produced with just two known sequences. So you can see that the outward facing areas are not very stable. So different portions of the protein are going to have more or less predictive states. You can study them and compare them, and you can probably provide some general framework for how you think the folding will happen. But the longer the sequence and the less known the reference, the more opportunities for inaccurate predictions you will have. Okay, and thank you. Bobby's posted a link on how the virus ejects its RNA in the cells of the host. Okay, so in our next session, we'll pick up from where we ended up. Please have UCSF Chimera installed. We'll take a look at some of those relationships between sequence and structure. We'll try to answer some of the same questions that this publication answered and try to understand it from a biological perspective. Again, this recording will be made available, and I'll post all the information from today's session on the forum. If you have any questions, please do let us know, and we'll be looking forward to interacting with you again in our next session.